Okay. Thank you, Lior, for the introduction. I will present today the subject for my master thesis. It's regarding the number of human and bacterial cells in the body. I guess that some of you may have tackled this question of what is the ratio between the number of bacteria and human cells in the body. So let's do like a quick show of hands. You're not obliged to stay with your answer from the homework. You can switch if you feel like you got the wrong answer. So who thinks that the ratio is 100 bacterial cells to 1 human cell? Okay, so B, 10 bacterial cells to 1 human cell, so I think the majority. C, 1 to 1, so the other majority. And D, 1 to 10. Okay. I won't get to the E, I think it's enough. So actually, we in the lab really like to play with data, and this time we have a lot of data from your own homework results, and it's interesting to look at it. At least we think it could be interesting. So this is the histogram of the results you got for bacterial cell. So it's interesting to see first that the variation here is huge, right? It's like 16 orders of magnitude, which is extraordinary. But even if you forget about the outliers, you still stay with the bulk part is about 4 or 6 orders of magnitude, which is not bad. So I guess we would think that the outliers may be some error in calculation, some no, but all in all, we have still uncertain, let's say, about the question, but we also can look at the geometrical mean and the median, and you can see them here. They're about, let's say, about 100 trillion or tens of trillions. So again, for the human cells, here the distribution is a bit narrower. It's still, let's say, this 3 orders of magnitude for the bulk part, and interestingly, the median or the geometrical mean are pretty similar to what we've seen with bacterial cells. So this is fine. It's nice to look at this like this, but actually we have both answers for each of you, so we can also plot it on this scatter plot. And this is a log-log plot. This means the values of each axis are multiplication by this factor of 10. And who can tell me what are the diagonal lines? What does a diagonal line represent? Anyone?
Concentration.
Yeah, concentrations. This is because usually when you have a linear plot, the diagonal lines are constant, y minus x equal a constant. And here, because it's a log-log, y minus x is the ratio of y and x. So they are the ratios. And you can see here that most of the results were around these values we seen earlier of, let's say, between 1 trillion and 100 trillion. And let's say the correlation, there is no good correlation here, but most of the values of the ratio is between, let's say, 1 or 10 to 1 to 1 to 100. It was interesting for us to try and divide it by the background of each student to see whether, let's say, people from life science who may have heard about some consensus thought something one and other thing, but actually we found out that there are much too much life science students here, and it's really hard to get a signal. But it's also nice, and it's always nice to play with data. I think you get it from my lecture. Okay, so this was nice. Just let's conclude it with the histogram of the bacterial to human cell ratio. So you can see that like we said earlier, the main results are between 1 to 1 to 100 to 1, and the central deadnesses are around this, let's say, few to 1. Okay, so, and this all goes back. I guess most of you have heard of this consensus. It's taught usually in bachelor in biology. a consensus that in the human body, the bacterial cells outnumber human cells by 10 to 1, or at least 10 to 1. And you can see here few articles from like Scientific American, places you would assume would reflect the best practice. And it was interesting for us in my master, like to try and see where did this claim come from. And it was very interesting to see that everything goes back to one single paper from the 70s. So this is some example for the citation change. And you can see that there are very good journals like Nature and Science, and each cite each other. And in the end, it goes back to this paper from 77 by Savage. We himself used another paper of 72 from Lekki. We will go back to this paper later on. Okay, so we have this consensus, and now we understand that it all goes back to like one single source, and the question is whether it's true, right? And if we want to answer this question, we need to consider one important thing, and this is the question of accuracy or precision. So here we have a ratio of two variables, and each have its own uncertainties. That means that if we want to know the ratio, let's say by like a factor of 2 or like this kind of precision, we need to know each of this denominator and denominator. We need to know it by much better precision, right? Like if we know the bacterial cell number by an order of magnitude, and we know the human cell by an order of magnitude, the answer could have be wrong by 2 orders of magnitude or more. Like the error propagation is a bit more complex, but good. So now we understand that if we want to have like a good estimate for this ratio, we need to have even better estimates for both denominator and numerator. So let's begin our journey. So we'll start with the number of bacterial cells in the body, and for this we need to answer like where do bacteria reside in the human body, and this is like pretty well characterized that you can find bacteria throughout the human body. Actually, it's like on the outside of the human body. Even if you think about it, like the digestive system is also kind of outside of your body, just like your body is just like a donuts, it's like a torus, and the lumen of the digestive system is just like your outer of the skin is just outside of your body. So you won't find, like in healthy condition, you are not supposed to find bacteria within the tissues or within the body, but like throughout, all over the body, you will find bacteria, mainly in the skin and all the like mucosal organs that has like outage, and you can see here some of the places. But okay, it's not enough only to know like where does the bacteria stand. We need to know something more quantitatively, right? We need to know something about the distribution. What can we look at when we try to know something about this distribution? Like one measurement, we might have heard about it in the last lecture, let's say, which was on the surface area and the concentration. Yeah, let's talk about concentration, right? Like if we know the bacterial concentration, we might have some estimate for the distribution of bacteria throughout the different organs of the body. So this is really what we've done. We've gathered from the literature the concentration for the different organs, different systems, and you can see different parts of the digestive system, because we'll see in minutes it's a bit important. And this is interesting. Only like this numbers are number of bacterial cell per milliliter or of like the content, and except for the skin is density of surface area. Okay? So you can see that like the largest or the highest concentration are in the dental plaque or in the mouth and in the feces or in the colon, large intestine, the end of the digestive system. Okay, this is nice, and we can combine this with estimates for the volume of the different systems or the area in the case of the skin to get an order of magnitude estimate for the number of bacteria in each of the system. So we see here that all in all, the colon bacteria dominate everything else by at least 2 orders of magnitude. This means that, like, if we want to have a good estimate for the bacterial cells in the human body, we might focus on the bacteria in the colon, and everything else will, like, move the needle like 1%, few percent. So it's not the level of precision we are looking for, right? We won't know everything by 2%, so it's okay just to focus on the colon. So far, so good. Question?
I have a question. Like, in the colon, is it really a matter of volume? Is it not more a matter of surface area? Like,
yeah.
Bacteria swim.
So I will get more into it then, but actually, yeah, it's volume. It's like the bacteria are throughout the content. They are not concentrated only in a thin layer.
Yeah, I mean, so it doesn't have to be only. It has to be like orders of magnitude different. But if
yeah. So let's in a minute we'll talk about different strategies how to estimate the number of bacteria in the colon, and we'll compare the different strategies we can try to use. You can. If it won't answer, let's revise it. So, just like to recap, so we know bacteria are all over the body, but we understand from the concentration that most of them are going to be in the colon. So we have an order of magnitude estimate, but we want to have something that is more precise, and this now we're going to get to this. Okay? Okay. As I promised, so let's talk. How can we how can we estimate the number of bacteria in the colon? So the first offer is suggestion is let's look at the ratios of surface area between the colon and that of bacterium. Some of you did it quite nice in the exercise, but there are few things we need to know in order to use this method. We need to know something about how much of the bacteria adhere to the wall, how much of them don't adhere to the wall, like how this model of only bacterial that are sitting on the wall of the intestine is a good model. We need to know the surface area of the colon, and we need to know layers. Like if you think about this model of, we need to do something about how much layers of bacteria are adhering to the wall, right? Like it's not necessarily only one layer, but this won't give us a good estimate. Okay, so this was one method. Another method is to look at the volume and look at the ratio of volume between colon and that of a single bacterium. So for this, we need to know how much of the volume is made out of bacteria, right? It could be that only 1% of the colon content is made of bacteria, but it could be 80%, and this will be a large factor. We also need to know what is the mass of an average bacteria. So most of the bacteria in the gut are not E. coli. They are anaerobic bacteria. We find them very hard to cultivate and to grow in the lab. So we don't have good intuition really to what are the real bacteria we have in the gut. So we'll have to get some measurement of this. And the last method is to use, similar to what I've shown earlier with the solution, is to lose concentration and the content of the colon. And here we need to know two things. We need to know what is the volume of the colon's content, and we need to know the concentration of bacteria. Okay, so in our research we used the last method, and we think it was like the best because we had this kind of information and we had good data. Like if we had good data for the other methods, we might have used them. Okay, let's go back to Lekki, the source of all events, the source of this very, very cool back-on-the-envelope calculation. He did it in the 70s. He's like a very interesting guy. I think they gave him an honorary samurai something from a university in Japan. It was like a fascinating character roaming around during the 20th century. And he did a very cool back-envelope calculation, much like the one we like to do here, or we did in our first exercise. And he did it very similar to the method I suggested. He took the concentration of bacteria and he took the volume of the gastrointestinal tract and he multiplied them both, multiplied it by the density of water, and he got an estimate of about 10 to the 14 bacteria. So it's a very cool back-envelope calculation. Some problem with it is that there is no sources. It just like came up with these numbers. Might know them because he is dealing with it, or I don't know. And another very, very important thing is that he multiplied by the volume of the entire gastrointestinal tract. And if you remember, I think like 3 slides ago, we talked about the different concentration of bacteria throughout the digestive system, and we saw that in the stomach and in the small intestine, the concentration are at least 3 orders of magnitude less than in the colon. So this means that taking the volume of the entire digestive system wouldn't be true, right? It wouldn't be, it will be overestimating by a factor of, let's say, 3 or something, or even more, which might not be bad if you want to get an order of magnitude, but if you want to be more precise, it's not the best. Okay. So how can we know the volume of the content of the colon? Some of you use this in your exercise, and I'm sure you had a bit trouble with it. So first we can try and using geometric measurement and compute using this very complicated formula of volume of a cylinder to get to this estimate of about, like, let's say, about 1.5 liters. And this is quite a lot for the volume content, and this is because these geometric measurements are measured from cadavers, and in real life there is a lot of contraction of the muscles of the gut, and some of the volume of the cylinder isn't filled at one time. So it's like a nice upper boundary for the volume of the content, but it's not really representative. There were other methods throughout the years used to try and get a better estimate for this volume of the content. So some measurement of post-mortem, like people who sudden death in an accident or something like that, you can actually measure the content if it's close enough to the time of death, or flow measurement of different parts in the digestive system. Some scans using X-ray or MRI, and when we integrate everything, we get to an estimate of about, let's say, about 0.5 liter, 400 milliliter. And we used this very nice paper who used all these kind of measurements in the 60s and got this estimate. And we also integrated with relatively new data from MRI studies. And it's interesting, they got that the volume of the content is linearly correlated with the height of the person. So there are a few other details here. There is another study, but it's like tall Danish man. You need to scale it, and all in all, using this, let's say, two main methods. This paper with all this kind of measurements and the MRI, we get to a total of around 400 milliliter. So there is a pretty nice way to sanity check that, or at least get a lower bound that it's not so far from that. Can anyone think of kind of this method? Yeah.
How much do we dispose of?
Yeah, that's
Go to the toilet.
Yeah, that's a great idea. So apparently, when you do this kind of research, you find that people research this kind of things, and it's a bit messy. So apparently, an adult person in Britain in the 80s, from the cohort of these studies, he produced something like that, and the colonic transit time, the time that it takes from the when it gets to the colon to get out, it's about a day and a half. And when you take these things in account, you get something like about a bit more than 200 grams. So this is a lower bound because in the entire content you also get some waters that are observed. And actually there is apparently this also really depends on the diet, like a high fiber diet really increase this volume. So it's, but it do gives us a sense that we're in the right place. Okay, this is what sanity checks are for, to see that we are not making a huge mistake. Okay, another cool thing like to think about is that each bowel movement reduces like the whole content of the colon by, let's say, a quarter or a third. So this is something we should like take into account when we consider the precision and the total, like the end results, the variation, the dynamic variation throughout time is pretty large. Okay. So bacterial concentration, up until now we talked about the volume. We saw that instead of taking 1 liter like Lekki took, we should only concentrate on the colon, and the colon is only about 400 milliliter. Now we need to know something about the bacterial concentration. And here we gather 14 different research, and you can see like the distribution. This is a linear box plot, and this is a logarithmic box plot. You can see that it's distributed around this value of about 10 to the 11 bacterial cells per gram or per milliliter. It's about the same. And this is it. Okay. So let's summarize this. So we've seen Lekki estimate. He used this 1 liter and this number of 10 to the 14 cells per liter to get this order of magnitude. But now we understand that we should only focus on the colon with a lower volume of about 400 milliliter, and the concentration are about right. But now we know it with like a reference and not from. Okay. So all in all, about 40 trillion bacteria. And you can see here the use of the different tildes Ron talked about in his last lecture. So in Lekki's estimate, we are doing a back on the envelope order of magnitude estimate, and we use the tilde, and now we know this like by much more precise, like tens of % of uncertainty, not a factor of 3. And we use the double tilde. Okay.
Have you tried also to actually calculate what is the actual variation? Like if you're taking, I don't know, the guy with the less amount of bacteria in his body and the guy with the mass. Like
yeah. So
what would be the range? Like how many orders of magnitude we spend?
So in the paper we also try to give also the variation, the confidence of variance, like the CV, what it's called, and it's about I think 50% of the end result. Like it means that the standard, it's like the ratio between the standard deviation across the population and the mean. So it means that the standard deviation is about half of the mean, and I think it maybe for the end result and not for this specifically, and you can find it here. And we also gave an estimate for the uncertainty of the mean. Right? These are two different things. They're both interesting. Like, what is the variation across the population? We saw that we are looking at a specific guy with this concentration, but the concentration might be a bit higher. The colon might be a bit smaller. Right? So we also want to know what is the level of precision, because our estimates are not precise. But we also want to know the variation across population. We try to give both in the paper. You can find them there. Okay, back to you guys. We talked about how many bacteria are in the colon, and now I'm going to ask you, what do you think is the total mass of the bacteria in the body? So about 40 trillion bacteria, right? So who thinks it's A, about 50 grams? Who thinks it's B, about 200 grams? So let's say about half. Who thinks it's C, 1 kilogram? The same half. And who think it's D, about 2 kilograms? Somebody think it's 5 kilograms? Can skip? I think so. So the answer is actually 200 grams. So how can we know that? So there was these two interesting scientists from the 80s that tried to take stool samples and divide them to the different, let's say, to the different components that make them. And one of these components, or two, were like bacterial matter. And by this method, they found out that about 55% of the dry matter is made out of bacteria. Okay, so this is dry matter, right? After I remove all the water. So what can this tell me about wet weight, right? We have I don't know, 400 milliliter of stool, and we don't, or colon content, and we want to know how much of them are bacteria. So anyone has an idea?
So dry mass is supposed to be around 30%.
Okay. And
that's it.
For both
okay.
bacteria
Yeah, so the water content is quite similar between bacteria and colon content, right? And almost every other biological system we work on, right? It's about 70% water or 75% water, and you can see here like the different bacteria might be different, but the ranges are between like 60 and 80%. So let's say that all in all, it's induced some uncertainty, but we can say that the bacterial mass is about half of the wet content, which what we used to work with, and it's nice. This is understandable? Okay. So now if we take 400 milligram, around 400 milligram of stool, and of content, when we multiply it by 50%, we get 200 grams. So this is cool because previous estimates or previous sentences are saying something like 3% of your body is made out of bacteria, and you can say that it means that kilograms of your body are bacteria. But if you remember now that the entire digestive system is like 1 liter, and where is the bacteria are, it's about like half a liter. So it just doesn't fit, right? So it's only a few hundred grams. Okay.
One.
Yes.
Where is the number 400 grams of stool comes from? Because
No, so it's not stool. It's like colon content. It's like there is like let's say a philosophical question of where does your colon content become stool, because it's like pretty much the same, right? Okay. So now to a very interesting idea we came by when we dealt with this, and this is like, think of a version of Ron's book, but for children, and it's all about poo. So this is a very nice, this is also here, it's a very nice book written for, like a scientific book for children. It's half humorous. It's very funny. It's also built from vignettes, and they're all surrounding the digestive system and poo and interesting stuff. I will pass it, and you can. So if some of you prefer to read a vignette of this book instead of Ron's book for the next exercise, you have my permission. Okay, so when they were writing this book, Benz, one of his authors, sent us a question by email when he saw our paper, and let's say like the gist of the question was, how come about 3/4 of the content is made out of water, and about half of the content is made out of bacteria? So let's just someone tell me that show me that.
It's also part of the water.
Yes, right. This is like and there is like this a nice diagram they made in one of the papers. Also, they got something also wrong there, but never mind. So this is cool. Pass it on, and so let's conclude this part. Okay.
So just for the calculation of how much does it weigh, you estimated the number of bacteria in the body. Why not just multiply it by the weight of one bacteria?
Okay. So how big? Like, what is the size of one bacteria? This is like the other way around, right? Like, if everything was E. coli and we would known it's 1 picogram, we actually could have multiplied this 40 trillion by 1 picogram, and we'll get how much?
40 grams.
40 grams, right? So, but it turned out it's not like that. But we can do like the opposite now. We can use this value of 200 grams, or about half of the wet weight is made out of bacteria, to calculate the size of single bacterium, right? And you want to do this, okay, let's just use this formula. So actually, if you think about it, you can just like multiply by 400 milliliter in the both numerator and denominator, and it will make a bit more sense, let's say. Like it's 200 gram divided by 40 trillion, so it's 50% divided by 10 to the 11. Make sense?
Again?
Okay, so we'll stop like for Tush.
You're
Just for board.
Okay, so let's. So let's do it like this. So we say. איפה שחור? אני לא יודע אם הוא עובד. So, איפה שחור?
The green one is better.
I think we have one more. Yeah. Thank you. So what is half? Half is the bacteria mass, כאילו mass bacteria, let's say, חלקי, divided by the content mass. Right? And we can say if we divide it, let's say we multiply it by the concentration. So what is the concentration we've seen earlier? It's, let's say it's, okay. We want to have this kind of, we want mass of bacteria divided by number of bacteria, right? And we want the content to cancel out, right? So we can multiply by this, but what is this? This is 1 over the concentration we've seen earlier. Is it? So we've seen earlier, it's like 1 gram for 10 to the 11 bacteria, right? We know this. So actually what we've done here is like multiply by 1, right? And now this is, and we have 1 gram, 11, something's wrong. Okay. Let's redo it. I think it's more easy like this way. We know there are about 200 grams of bacteria, right? And there are about 40 trillion bacteria.
Power 12.
Well,
The power should be 12.
the power should be 12, you're right. So let's do the other round. I'm sorry for the confusion. 40 trillion bacteria divided by 200 grams, right? This is true. So it's cancelled and it's 10 to the 5, 10 to the 12 gram per bacteria. Okay, sorry for the confusion, but actually, if we divide both of them by 400,
It's not 5, it's 1 divided by 5. It's 0.25.
no, it's
40 divided by 200 is 1 divided by 5.
bacteria.
Is like 2 to the 10 to the 11.
What? אני עדיין בבקטריה פר גרם?
And 1 gram per bacteria.
וואי, זה לא לעולם... I'm just getting it wrong, sorry. Okay, דווקא, okay, it was okay the other way, sorry. Okay, let's do it all over and it will be okay. I'm sorry for the confusion. So, on top, we want to have bacteria, we want to have gram, and on the bottom, we want to have bacteria, right? Okay. So 200 gram divided by 40 trillion bacteria. And this is 5 multiplied by 10 to the minus 12 gram. Okay. This is it. And what I tried to say earlier is that if we're dividing each one of them by 400, right, what we get is, like we got here half, which is the mass of bacteria divided by the total mass. And on the other eye, we have like the concentration. 10 to the 11 bacteria per total mass, which is the concentration. Okay. I'm very sorry for the confusion. Maybe let's do a break.
So, 10 minutes?
Okay, let's continue. So far, we managed to have let's say, a good estimate for the number of bacteria in the human body, and even know something about their size, failing miserably on the way. So let's talk about the number of human cells. Okay, so I want someone, volunteer, to tell me, to tell the class, how did he calculated the number of human cells in the body? Okay, yeah.
I just googled what is the average volume of a human cell, and then estimated what's the total volume of the body of like a human, and then just divided it.
Okay. Did anyone, like everyone heard? Just like, I will reiterate it using this figure from Ron's book. Some of you might seen it. It's just like we are taking the mass or the volume. It's like easy to switch between them of the total body, and let's use a rounded number, and we take the volume of a characteristic human cell of about between 1,000 and 10,000 micrometer cube, and by doing the division, we get to this order of magnitude estimate. Right? This is very cool estimate, and it's nice example, I think. Okay, but what's the problem? What could be the problem with this kind of estimate, or this kind of method? Shoot. Rude.
That you might have order of magnitude difference in the size of cells, and therefore you can have like kind of like few kind of cells which are like, if you have, for instance, you have like one population of cells that is 3 order of magnitude smaller, it can just, in the terms of number, it can be just outweighting all other kind of cells in the body.
Okay, so the varieties of cell sizes could have a big impact on this, and we'll get like more into the details of what you just said.
But it really comes down to how accurate you are, like how, if the characteristic cell is actually the mean, and it should represent all the variability. We need only
Okay,
to
but
mean
it,
the
so
mean mass.
okay, so this is like, let's say you nailed the two main points I wanted to say. This is like, what is the accuracy? Like we've seen, it's like order of magnitude accuracy, and we need something more precise. And also, what is it really that the representative or the characteristic cells are really is the mean cell, right? Like, it's really, the question is very different. Like, when the characteristic cell is much more like, when we think of a human cell, what is this cell? Or when we do something in the lab with human cells, what is the cell that we are using? But it might be, and we might see it in a minute, that the characteristic cell, that the HeLa cell, or I don't know, whichever cell you think about when you think about human cell, is not really the mean cell. It doesn't have the mean size. Right? And this is really goes along with the what Or commented. Okay. So as Or said, there is a very large varieties of cell sizes in the human body, and this is a table from Ron's book, and I think from the exercise I have checked, so at least say a quarter of you read this vignette and know this, but it's a very cool vignette if you haven't. So you can see here a table with different cell types and their different volume, and you can see that it's spread like at least 3-4 orders of magnitudes within like the main cell types. And this, as Or just said, it's create a problem, because if we have such a huge difference, it might be that there is a very large population of small cells, and they dominate everything. It's just like if we have, I don't know, like a vessel, and we put there a lot of rocks from different sizes, and some of it is just like a very small griddle, gravel, and then you will have like, and if you just like enumerate every rock and you enumerate the gravel and you give them the same number, it will be filled with gravel. It might be, right?
The weighted mean, right?
Yeah, because you need the weight. Okay, so and this is another depiction of this variety. It's like a cell mass axis. It's similar to the axis Ron used, and you can see here also E. coli and bacterial like representative cell, and how does it compare with other cells from the human body. And you can see that the human cells, they vary like large orders of magnitude. Okay, and this is some icons of different cells. Okay, so can you give me an example of a small cell that might be easy to calculate?
Red blood cell.
Oh, nice. So, okay. So let's do it on the board together. But I promise this time it will be better. So, who can think of what do we need to know in order to calculate the number of red blood cells? Concentration, nice. What else? Blood volume. And is it enough?
Yes.
Yes, it's enough. Nice. So, anyone have taken a blood test recently?
Mm-hmm.
And you remember his blood count, red blood cells count? So, actually, I've taken like a few years ago, maybe 6 years ago, and here you can see like some of my, it's my personal information. I'm okay with sharing it with you. So you can see here, red blood cells, 4.75, so let's do it 5. What are the units? It's millions per microliter. It's okay. So 5, 10 to the 6 cells per microliter. Okay, so what is the volume of an adult man, let's say? Volume of blood of adult. So,
So 70
so it's about 5. It's about 5 liters. There are a lot. It's when you have something that you can measure, you can find like a lot of different data and a lot of different, let's say, equations you can use to estimate this. But it's around 5 liters for an adult man of a height of about 170 centimeters. So, right, we multiply it by 5 liters. So let's multiply by 1. How many microliters are in a liter? Right, 6 microliter. And so what is the result is 25 trillion red blood cells. So this is quite a lot. We've seen earlier the range was between 10 to the 13 and 10 to the 14. So already we are in the middle of the range and we talked only about one cell type. Does anyone know what is the mass of a red blood cells? We've seen it in what is the other one? So actually you also can get it from like a good estimate for your blood test. This is what you called, I think it's mean corpuscular volume or something like that. This is this is around the volume of the mean volume of red blood cells in your blood. So it's about, let's say, 90 femtoliters. So let's use 100 femtoliter, which, if you remember from what we did with E. coli, it's about 1 femtoliter, 1 picogram. So it's around, so the mass is around 100 picograms. Pico cube. I'm sure there is something. So, okay, so what is the total mass of the red blood cells in the blood? So we have 25 trillion cells, and we have, let's do already convert it to 10 to the minus 10 gram per red blood cell. So all in all, it's about 2.5 kilograms. Makes sense? Okay, and I don't know how many of you know this, but about half of the blood is made out of red blood cells. This is known, like half of the blood is red blood cells, almost half is plasma, and the white blood cells are something like 1%. So this is very cool, and it shows us what Or just told us might be the problem, right? We have only 2.5 kilograms of red blood cells, but they already make up something that is equivalent to the order of magnitude we've seen earlier. Okay, so this is a version of not me on the board doing it. So how can we have a better estimate? Any ideas?
We know the abundance of each cell, we can do the weighted mean.
If we know the abundance, right? And if we know the abundance of every cell, we know the answer already, right? Okay, but it sounds like a but this is the best method we came up with. It's just like what we call an inventory method, in which we go we take the main system, different organs of the body, and for each we try to enumerate the number of cells using consideration as we see now with the red blood cell. It's much more work from what we can do with like only dividing two numbers, but it could give us a much better estimate.
You can kind of rule out any cell which is heavier by more than like a factor of 100 than red blood cells, right? Because it's already you have 2.5 kilos, so everything that is
you're right.
Even an order of magnitude will almost demolish the contribution to the amount.
Your intuition is good if we want to answer only the question of what is the total number of cells. But if we are doing this work, we might also want to know something about the distribution. We want to know, we want to make sense. We want to have the ability to check ourselves with different considerations. But you're right. If we want only the number, we might look at this mass axis, and everything that is like 2 order of magnitude bigger than red blood cells, we just know it won't change, and we are not even going to consider it. You're right. So there are different ways to estimate the number of cells in different tissues. We'll see some examples, but we use whatever data we can find from the literature. Sometimes we can find people who try to find the number by themselves. See it in a minute. And sometimes we need to integrate a lot of different details to know something about the density or the DNA content and stuff like that. Okay, so we did this and we got a pretty good picture. We were close to have like a good estimate for the total number of cells and the distribution. And then we got scooped. A group of Italian doctors published this paper. It was just when we, in the middle of our work on this content, it's a paper that did a very similar thing to what we try to do like an inventory method. They worked out different cell types and tried to get their numbers. Although there were, let's say, at least few caveats in their work. One of the caveats was that they was very, very bad at like representing their work. There was no visualization of any kind in their work, and this is like the most basic visualization, and we did it to their work. And actually, with their numbers are also a few problems. So we decided to show today, like, to go by the main factors they find. They found only, you see here, I think it's 6 major contributors, and everything else is like 2% or something. So let's just look at, okay, sorry. This is a table for this 6, and you can see their estimates and our estimates, and I'll just use it to, like for some example of how we estimated different things. So red blood cells we have already seen. I think I need to click. So let's talk about glial cells. Glial cells are the non-neuron cells in the brain, most of the non-neuron cells in the brain. I encourage you to go and read about them, they're interesting. So apparently there was another very famous 10-to-1 claim, and this was that in the brain there are 10 glial cells for every neuron, and this was believed, or at least famous. And this group of doctors, they used this claim with no really good reference to calculate the number of glial cells, and they got it wrong by about a factor of 10, because this very cool study from 2009, they just got to enumerated the different densities and different numbers of glial and neuron cells in the brain. You can see here in their picture, you can see it in the different segments of the brain, and all in all it's around 100 billion neurons and glia each. Cool. Here we only found in the literature the number. It was nice and easy, or not as easy. Nice. So another example is in endothelial cells. So endothelial cells are the cells that line the walls of the vessels, different blood vessels. There is only one layer of these cells. This is important. Like in every, it doesn't matter what is the thickness of or how, how important is the vessel. There is only one cell. This is like the vessel that touches the blood, and there is only one cell. So you can calculate the number by, if you know like the total surface area of the different vessels in the body, you can divide it by the surface area of a single endothelial cells and get their numbers. But this number is of the total surface area of vessels in the body is not really easy to come by, right? You have to calculate it somehow, and they didn't really calculate by themselves. They took it from somewhere. So in our work, we try to do it like in a more rigorous way, and we look at the different type of vessels, the capillaries, the small veins, and for each we calculated their volume and their surface area, and we used the same logic to have a better estimate of the total number of endothelial cells. And what is nice is that now we can say that let's say, most of the endothelial cells are within the small veins and the capillaries and not in the large vessels. So this is a nice and more thorough way. I have something else. So the last example is dermal fibroblasts. Dermis is the second layer of the skin. It's mostly connective tissue. Fibroblasts are the cells that like, emit the matrix. And Bianconi used this density that was measured by some study, and they multiplied by the surface area of the skin, but they somehow, like, I don't know, missed two orders of magnitudes in their unit conversion, like something with millimeter, centimeter square, and they got it wrong by 2 orders of magnitude. So if it happens to you in an exercise, it could happen to these guys in a paper, but it shouldn't. So we did this a bit better, and we also separate two different area of the dermis, and we got a better estimate. Okay. All in all, we got this very, very cool distribution of the different cells in the body, and you can see here there are about 30 trillion cells in the human body. About 84% of them are red blood cells. So it wasn't a coincidence that we got this earlier, and it's interesting. Other major parts you can see here, other cells. So all in all, about 90% of the cells here are enucleated. They don't have nucleus, so it's interesting. Also, it's a philosophical question whether to think of platelets as cells. Are they really cells? Are fragments of these megakaryocytes? So it's also a discussion whether to even consider them.
How is it different when you're comparing to the, not the amount, but the weight?
Oh, very, let's say, wonderful question. So so the question is, how can we check ourselves? And the answer is, we can try to use weight and to have some sanity check on this. And so before we look at this, just let's say how can we derive the weight? So in a 70 kilograms man, there are about 46 kilograms of cell mass. This also depends on who you are, on what is your how do you call it? Like the percentage of fat in your body and other stuff. But this is around, and also you can think that every tissue has its like different structure. It could be that it's filled with cells, like 90% of the mass of the tissue is cells, and some other tissues might be like much less. So there is a very cool way to use, like to infer the cellular mass of tissues. You can find it in the paper if you want to, but it uses like the potassium concentration in the different tissues, like to estimate the percentage of the mass that is in a cell. If you remember Ron's lecture about concentration, we saw that potassium is really, really strongly kept at certain levels inside cells, and out of the cells it's really low, so it's useful. For example, in skeletal mass, there's about 20 kilograms of cellular mass in like from 28 kilograms of the tissue. So this is all details, just to come with this nice picture, juxtaposing the number of cells and their mass. We much less segmentation here. We just can see that in terms of numbers, you have the red blood cells that dominate everything, but they're only about 2.5 kilograms, and you can see that most of the, like 3/4 of the cellular mass is concentrated in muscle cells and adipocytes, fat cells, although they make up like 2 promilles of the total numbers. So it's also interesting to put the bacteria here, which we've seen earlier. There are about 40 trillion bacteria, and they only weigh about 0.2. So it's even more to the extreme with them. So, okay. So we sanity checked by mass. So, but did we find that we miss something. So going back to Or's comment from before, great comment. So now we know that we kind of have the total mass, and the precision here is like, let's say, it can't be that we missed by few kilograms. It's like at least maybe 1 kilogram we missed, or maybe, but not more than that. And we know the overall numbers are about 30 trillion. So that means that if we miss something, in order to make any change, to move the needle, it has to contain at least like 1 trillion, because if it's like 30.3 trillion, it doesn't really change our estimate. This is the level of precision we have. And it has to weigh like at most 1 kilogram, right? So this make us like, now we know which cells we need to focus on. It's only small cells, and only cells that are less than, let's say, the representative or average cell we like to use. And we found, when we did this work, we found one good candidate. This was like the white blood cells, and especially the lymphocytes. And we've seen that in the previous work, they accounted only for the blood cells in the blood. But blood cells, we call them blood cells because they're in the blood, but they are in every tissue. They're responsible for most of the immune system. And actually, when we, at least for lymphocytes, or kind of white blood cells, we've seen that there are about 500 billion of them. This from this paper from the 70s, and they weigh about 300 grams. Okay, and actually now over the past, I don't know, like a semester or two, we are trying to go like more deeper into this issue of the immune system and look at how the cells of the immune system distribute across different tissues and try to better this estimate. But it's like in terms of the total number of human cells, it's still quite the same. Okay, so let's wrap it up with going back to the bacterial to human cell ratio. So we've seen the bacteria side, Lekki's estimate, 10 to the 14, and our estimate, revising it, knowing that we need to focus on the colon, we got about 40 trillion. Now the human cells part, they only took this value without any primary reference, like just from a textbook or something. And now we know it's about 30 trillion, and we can remember that by remembering the calculation we did on the board of the red blood cells. Cool. When we submitted this paper, we got a very nice review, really very nice review, but the nicer reviewer gave us this comment, and you can read it, but I just like what it's pointing at, that up until now we only talked about the reference man, like a 70 kilogram adult, like all the values we took were from this, for this, but what about other population? How does this ratio, how does the number of different cells are changing when you look at women or infants, elderly, like with obesity? Okay, so how can we generalize these values we got? Does anyone have any idea?
Extrapolate over demographics.
Okay, how do we do that?
Well, first you need to choose a country, probably, where
No, let's say we know what demographic we want to use, but what do we need to know in order to extrapolate our results? See what?
even you know how much an average 2-year-old weight, for example.
Yeah, let's say I know this. So how do I extrapolate?
You divide by 17. Hope it's a linear.
So you say I just extrapolate from the mass. But the problem is that we have a ratio here, right? We have a ratio of human and bacterial cell. So if I extrapolate each linearly by mass, they will just cancel out, and I will stay with the same ratio. So I can say, like, with a first order approximation, it's the same for everything because everything is scaled with mass, if it's true. Okay, other idea. This is a nice idea.
It's for a general.
Yeah. Other ideas?
You might want to look at how the diet in different regions affects, say, the population of bacteria inside the gut.
So I will look at how different segments I want to extrapolate to, how the different concentration in the gut looks like, if I know. Okay, that's important, but is it enough?
No.
Okay.
I don't know if there's a very simple way of doing it for some populations because, for example, a newborn baby is thought to be sterile in terms of bacteria, so you think it has minimal bacteria, but also the number of cells are like vastly different ratio-wise to an adult. And also with women, obviously women are built with certain differences that I'm not sure you can get just by scaling or.
Okay, so Dina, as I understand, Dina says that it might not be like a linear extrapolation using mass or something like that.
I think you can try. Most of the things are linear to some constants. You can determine which are the big factors that are linear with, like the concentration and the whatever. You can start 4 or 6 factors like this, and then you can have like a simple model.
Great. What is your name?
No.
No. So Noam made a great point. That's it. We can look at our estimate and try to determine what are the major factors in this estimate. And for each, now we have much less, like we are not gonna do everything we did here for this infant or obese or woman. And we just look at these major factors, and for each we try to understand how does they change, or if they change between, and that might be linearly, might be not, might be, and we can do this. And this is really what we've done. We found that if you look at this, this is our estimate, right? This is gives the main bulk of our estimate. We have here the volume of the colon and the concentration in the colon, and we have here the volume of the blood and the concentration of red blood cells. And these are 4 factors that if we know them, we have pretty good idea of what is the ratio, right? At least for the what we want to know. And this is a very good, this goes with what Noam just said, that might be a good factors we can use to extrapolate to other populations, right? Because we know what are the main factors in what we've done, we can use it to have an idea about everything else. So these are really the factors. And so now we will use reference for an adult woman, for young infant and elder, and for an obese. So each has its own it's not really it's not necessarily extrapolated by mass only, okay? Because an obese, so it mainly is, I don't know, adipose tissue is more than in non-obese, but what does it mean on the blood? We can try and look for that and okay. So we have these tables that compare them. So this is the estimate we have for the reference man. For reference woman, we see that the main things that change here is the red blood cell concentrations and the volume of the blood. They are both smaller. The volume of the blood is mainly because women, the reference woman on average is is lighter than the reference man, but the concentration of red blood cells, it's a known thing that women have less red blood cells, they have less iron. We can talk about it.
And the colon is the same.
So the colon content, we saw that okay, so if you remember the MRI study, so there is an index between height and weight, and it seems that, at least from these studies, and it's not it seems that the index for women is a bit higher than the index for men. So you get for the different heights that women are a bit higher, but it's not such a big change. And you have also the result for infant, elderly, obese. It's interesting to see that we have an idea of how different things change, but the concentration of bacteria, it's really hard to find any data to suggest any change. It doesn't mean that there is no change. It means just it wasn't really well characterized. So here we are using the same value, And this is something that is a caveat, and we would love to have a better thinking. So, but all in all, it's interesting to see that everything is like up to a factor of 2. So it's not bad. Like even it's not, the uncertainty of our estimate, it's just like the variation across different populations. It's not so different from what we did for a man, and I think it's interesting.
Well,
Yeah.
can you talk about the blood volume, really the differences?
Blood volume?
Like the obese one?
Yeah, so it's it's not a very well characterized, but it seems that there are models of what is the blood volume when for obese. It's like one study we managed to find, and it says that we thought that it's mainly the adipocyte tissue, but apparently the blood also there is an increase in the blood volume like to support this.
How can it increase?
What?
The volume of blood.
More
The-
blood
the-
is more vascularization.
So more vascular.
Yeah, you have. Okay, so we'll end soon. So some knowledge gap. Okay, the אמור להיות. It's supposed to be how well, not wow well, but let's say wow, well. So there are some knowledge gaps in our estimate. We are not trying to hide them. There is a question of how well the fecal bacterial concentration represent the colon. Like, does it represent well like the entire colon, maybe like the last half of it or whatever. We've seen that there is some data about column volume, and we have some indexes, but it would have been great to know it better. We've seen that we saw some effect on different populations, but it could be very interesting to look at different ethnicity, geography, but it's not necessarily that we have the data, right? We've seen that for the concentration of bacteria, we don't have good data for to segment this, but maybe there will be, and it could be very interesting. And there are also the prediction of the bacterial, the average mass of bacteria, which could be interesting if someone managed to check it. Okay, some take-home message before I go to a teaser for next class. So I want you to get out of here thinking that the back-envelope calculations are really great and they're very useful, but we need to know when to use them and when are they enough and what claim can we do with them. And when something is getting to this realms of being a common knowledge, it's something we need to have a bit more, let's say, precision about, and to know it a bit more, a bit well. And this is, we've seen this example, I think, very strikingly. And another thing is that you've seen these citations, and there are thousands of citations that goes back to this tree that we've seen. It's just like more than 3,000. And it's very interesting, like how this field of microbiome that really is in interest, and they use this in their introduction because they want to mention bacteria as something important, and they just use this value, like Ron said, they use numbers as ornaments, and they don't really think of what this number means, and they don't really try to understand what they come from. And I think at least the lesson we need to learn here is that if we use this kind of numbers, try to see what is there behind them, what is the primary source they come. Like, it's always a good idea if you use a number and you really want to use it to go back to the primary source. Sometimes you find out that it's just like a textbook that has no really meaning, and it's a question of whether you should use it or whether it's. And the last thing is that by asking what is the dominant factor, we can like extrapolate our results. We can make sense of more than the focal point on which we look at the study, and this is like a good example. Okay, so a teaser for next class. So next class is going to be about rates, and we know that in the cells in the body are being replaced continuously. So up until now, we look at the body only as like a stationary model. It's like it just exists, but we know that like the time I'm here, my cells are being regenerated, are some of them dying, and actually this is just like the ship of Theseus. This is like the Greek mythology story about the hero of Athens who goes on a very long journey, and throughout this journey, like whenever a plank of his ship is broken, he replaces it, and by the time he gets back home after years, he found out that the entire ship was replaced, and this serves as like a question of, like introduction to this field of identity and philosophical question of what makes something a whole, what makes something the same, and this also a lot of people like use this analogy with the human body and with sentences like every 7 years our body completely replaces itself. This also like for this philosophical debate, but actually we are interested in biology, and actually this is not true. We know that some cells in the body, they don't regenerate at all. Some other cells regenerate very fast. So this is not really a thing we can say, and we would love to change it and use a more quantitative statement. And here is your question, the teaser for next week, is, okay, so not the entire body replaced, let's just talk about one day, and what is the mass of cells that are being replaced each day? And I will give you 5 answers, and let's see. Who thinks it's about 0.1 grams that is being replaced each day? Okay. What about 1 gram? Okay, that's few. What about 10 grams? That's more than, I think this is half. What about 100 grams? That's few. And 1 kilogram? Okay. So this is a teaser for you. You are more than welcome to.
Do you include the bacteria in the?
No, not including. but nice try. So if you have any questions, you are also encouraged to stay and talk to us, whatever, if you want. It's an opportunity, and you can find the paper pretty easily by looking it online or finding it in our website, and if you have any questions.